Welcome everybody. This is the class on biotechnology, which is topic 20. You have already read some pages about biotechnology from the document that I gave you before the holidays, but I'm still going to go through all the slides with you so that you've got the complete class. So you'll see that in the third edition, biotechnology encompasses sections 410 to 414. In the second editions, the sections are a little bit scattered across the book. So you've got sections 2.5, 2.11 to 2.13, 3.29 and 3.30. All right, so here on the slide you can see some products of biotechnology, which maybe you wouldn't have thought of as such. Yeah, so we've got wine, we've got yogurt, we've got kefir, cheese, sauerkraut even, we've got beer, we've got bread. So these are all sorts of products that we can gain from what we would call traditional biotechnology. So traditional techniques that have used microorganisms to transform a product so that it is useful to humans or pleasant. Here are some more products of biotechnology. These are a little bit more modern. So you've got on the left hand upper corner, you've got antibiotics. Then you've got a washing powder, a biological washing powder. So not to be confused with a French bio, which is organic in English. A biological washing powder is a powder that uses enzymes to get rid of stains. Then you've got lactose-free milk, you've got insulin, which is also produced using biotechnology, you've got clear apple juice, which you may be surprised to learn is also a product of biotechnology. Then you've got tofu, which is basically a fungus grown, for consumption, obviously, but that's strictly speaking also biotechnology. And then you've got on the bottom left corner, you've got GMO corn. Yeah, so all genetically modified organisms are also a product of biotechnology. So what is biotechnology? So here's our definition. You've got the little pencil there, so if we're in class, I'd ask you to write down the definition. You can still do that, you can use this as if we were in class. So in biotechnology, microorganisms are used to help make products for human use. So that's either at home or in the industry. And so here you see a few pictures of traditional biotechnology, where people were not maybe even aware, or most certainly weren't aware, that they were using microorganisms. So bottom left, you've got a traditional dairy where microorganisms are used to make yogurt, cheese, and other dairy products. Then the black and white picture in the middle is people brewing beer. Then bottom right, you've got bread baking, which is also making use of biotechnology, microorganisms. And then the top right two images show winemaking and wine storage, which is, that has been known since Roman times, and that is also reliant on microorganisms, as you may know, but the Romans did not. So all of these traditional biotechnology methods, in all of these methods, people weren't necessarily aware that they were using microorganisms. They were just acting out of experience, and that experience was handed down as a tradition. Yeah, so now that we know about microorganisms, of course we can explain what is exactly going on there, but people at the time were using it thanks to experience without knowing what exactly was going on. So let's just go over some of the microorganisms that are used in biotechnology. So you've got bacteria, quite a lot of them. So I'm sure you all remember what bacteria are. There are prokaryotes, which means that they don't have a true, I hope you could all fill in the gap there. What do they not have? They don't have a true nucleus, and they also don't have membrane-bound organelles. They are single-celled, and they're the smallest living organisms. Now, there is a huge amount of bacteria on Earth. Yeah, so their total mass is at least 10 times greater than that of all other organisms put together, and that's a lot. Yeah, even just in our own bodies, there is an awfully great amount of bacteria. And so these are really very, very common, and if you know how to use them, they can be very potent allies in biotechnology. The good thing about bacteria is that they have very varied metabolisms. So what's a metabolism again? A metabolism, as hopefully you should know, is the sum of all chemical reactions going on in a cell. And because bacteria are so, old, they are actually also very varied. There's a lot of them. Evolution has produced a lot of different bacteria adapted to different environments, and they all have their very own metabolism. So that actually means that they produce lots of weird and wonderful stuff. And some of that stuff is really, really useful to us, which makes bacteria a really good go-to organism in biotechnology. And then you've got fungi, and among the fungi you have two big types of fungi. You have yeasts. So the picture at the bottom, these green cells with a yellow sort of scars on them, those are unicellular fungi, and these unicellular fungi, we call them yeasts. And if you are curious, the yellow marks on there, they're indeed scars. They're scars from budding. As you can see, right at the center of the image, you may see that there is a yeast cell that is in the process of dividing, and there's a little cell growing out of it. And once that cell is done, once the cell division is finished, there will be a scar remaining on the original parent cell. And then you've got molds as well amongst the microorganisms. So a little reminder here of the structure of a bacterial cell, just because it's good to remember that. You have to know what a bacterial cell looks like. Hopefully it'll ring a few bells, because this is something that you should have learned in troisième. So the main thing about bacterial cells is that they're very simple and they're very small. Maybe it doesn't look simple to you, but if you compare it to a eukaryotic cell, so those are cells with nuclei, this is very simple indeed. So the bacterial cell, it has a cell membrane. So in the cell membrane, we find that in every single cell. Here it's called, on the picture, it's called the plasma membrane, same thing. The plasma membrane is shown in yellow, and it surrounds the cytoplasm, which is the sort of jelly-like substance where everything is embedded into in a cell. That's the same for eukaryotic cells as well. So those two components, we find them in a eukaryotic cell as well. Surrounding the plasma membrane, you've got a cell wall, here shown in brown, which you hopefully remember that animal cells, for instance, don't have a cell wall. Plant and fungi cells do have a cell wall, but it's very, very different from bacterial cell walls. Okay, so this is something specific to bacterial cells. And then sometimes bacteria have this sort of slimy coat around them, which is here called a capsule, and that's the light brown layer on the outside. Then you have various other structures, very, very important. You've got the nucleoid, that's the purple structure on the inside of the cell, which contains the bacterial chromosome, which contains most of the information, of the genetic information of the bacteria. And then you also have a small ring of DNA, which is called a plasmid. So that is down bottom right of the bacterial cells, called a plasmid. And then you can see things like fimbriae, pili. Here you've got, at the top of the cell, there's this orange roddy kind of thing. It appears in its singular, which is a pilus. So the plural is pili. Fimbriae and pili are to do with cell adhesion. So how do cells stick to each other, and how do cells stick to the medium that they're growing on? And then you've got the very long spiral flagella, which are like the tail that can move the cell around for bacteria that can move. So that's your structure of a bacterial cell revised. There are different types of bacteria. Like I already said, there's actually millions of different types of bacteria, so there is no way that we are going to learn about all these here. These are three basic shapes that were recognized fairly early, once people had access to decent microscopes. But this is really just from what things look like. It doesn't tell you anything about what the bacterium does on the inside, what metabolism it has, or anything like that. Anyway, this is a classification which is widely used, so it's in your book as well. So on the left-hand side, we've got something called a bacillus, which is nothing else than a rod-shaped bacterium. Often the bacteria that look like that have the part bacillus in their name. So for instance, lactobacillus, which is a bacterium that digests the milk sugar, lactose. And then in the middle we've got a spirillum, which is a spiral bacterium, such as, for instance, Vibrio cholerae, which is the bacterium that causes the cholera. And then you've got a spherical type of bacterium, which is always called a coccus, such as Staphylococcus, for instance. Staphylococcus aureus, which you may know, can be highly resistant, or there are strains of Staph aureus that are highly resistant against antibiotics. And so that's how Staphylococcus is usually known. But yeah, now you know that it's a spherical bacterium. Yeasts. So I said it before, well, yeasts are getting the extra slide here though. So yeasts are single-celled fungi, and they're eukaryotic. Yeah, so once more, eukaryotic as opposed to prokaryotic, like bacteria. Eukaryotic cells have a true nucleus. So you can find that on this diagram of the dividing cell on the right-hand side. It's about to sort of divide between the two cells. The bigger sort of parent cell and the smaller daughter cell. That's the nucleus, the organelle that is sort of stretched out between the two. It's in the process of dividing. And eukaryotic cells also have membrane-bound organelles, such as mitochondria, chloroplasts, vacuoles, things like that. Hopefully you do all remember these different organelles and what they all do. Yeah? So the mitochondria, for instance, if you think mitochondrion, you should immediately think of the energy of the cell. The mitochondria is where energy is released from food, so that's where respiration takes place. Chloroplasts, which we don't find in fungi, obviously, are green and are the home of the photosynthetic pigment chlorophyll. So they are in plants. We find them in plants, and they're responsible for photosynthesis. Okay, an example of a yeast is Saccharomyces cerevisiae, which is the brewer's or also baker's yeast. So we'll see that this is the yeast that is responsible for brewing beer and baking bread. And you can see it on that picture. It's the same picture that I showed you on the previous slide. And very important to remember, yeasts are single-celled fungi, so one cell. Molds are multicellular fungi. They grow forming these weird and wonderfully looking hyphae. So hyphae are like cellular filaments, so long threads of cells that are all connected with one another. They don't form a body in the sense of an animal where there's like a 3D distribution of cells, if that makes any sense. But basically their whole body consists of threads, which is weird. So these threads are called hyphae. Molds, obviously, as fungi, they're also eukaryotic, and some examples are Penicillium and Aspergillus. So Penicillium, I think the name will already tell you what it does. It produces penicillin. So this was where penicillin. Oh my God, excuse me. This is the mold that we use to produce penicillin. On the picture on the right-hand side, you can see Aspergillus and Penicillium on a diagram, so a diagram of the two, and you can see that they're fairly similar, but that there are differences in their organization. You don't have to remember these specialized words, it's just to give you an idea of what they look like at the cellular level. Then if you culture Penicillium, for instance, in like a petri dish in the lab, it looks fairly disgusting. It has this characteristic green color, so this is it's also called green mold. And then you can also see what it looks like when Penicillium grows on an orange. Yeah, it turns green as well, and I'm sure you've all found oranges like that in your fruit baskets, and it's fairly disgusting as well, usually. Okay, so why do we use microorganisms in biotechnology? So there's a whole load of reasons, really. The first and foremost is that they're really easy to grow. Yeah, so you can grow them everywhere, even on your orange. You shouldn't be doing that, but you get the idea. It's really easy to have a microorganism as a pet. You don't have to, it's not terribly complicated to make them grow. Also, like I said, weird and wonderful metabolism. So they can make many different kinds of complex organic molecules, which can easily be harvested. So organic molecules are molecules relevant to life, such as proteins or lipids or carbohydrates or other types of organic molecules. Yeah, so there are obviously other types beyond these main groups. But the super amazing thing about microorganisms is that they make these substances and then they will secrete them into the medium that they're growing in, which means that you can harvest them really easily. So that's another big plus of microorganisms. What is very handy as well is that they have the same genetic code as other organisms. So I can take a human gene and tell a bacterium, such as E. coli, for instance, to make that gene according to my genetic code. Sorry, not to make the gene, to make the protein from my gene with that genetic code. And that is really, really helpful, that they're able to just read off human genes or genes from plants or other animals and make the same protein that we would be making. So that is quite amazing. Bacteria also have plasmids, which can be easily transferred from one species to another, from one strain to another. So they're a really good vehicle to carry genes around between different microorganisms. Now you can't do that with a mouse or something like that. So that's another big plus of bacteria. And also, speaking of mice, mice are fairly simple mammals, but still they probably have feelings and they have needs, and they can feel pain, and so you don't want to be using anything that can feel pain or anything like that in your biotechnology in the industry. So you can replace those by microorganisms which can be used without any ethical concerns, and that's a big plus as well.